Welcome to chapter 23, part 6, where we are going to begin digestive system physiology, which is one of the most fascinating things to me. I love digestive system physiology, but it is kind of intricate. We will have some charts for digestive enzymes similar to what we saw in the endocrine physiology, a little bit simpler than endophys, I think. And this is just a short recording to review some of the nutrient building blocks from way back in chapter 3. So first, enzymes. Enzymes are proteins, but they act as catalysts which accelerate the rate of reaction. That basically means that a reaction could happen, but it would take a much longer time, or actually the reaction might not happen if it never got enough energy. So these enzymes can kind of kickstart a reaction. And what's cool about an enzyme is that they can then be recycled and used again. Now, enzymes are very specific and they only act on a specific substrate. So a substrate is the target of an enzyme. And in this chapter, our substrates are going to be carbohydrates, proteins, and lipids. And what the enzymes will do is hydrolyze. So hydrolyze means break down using water. You'll also see the word hydrolysis. So lysis means to explode or break apart using water. And the building blocks of carbohydrates, the tiny forms of these, are what we're going to need to absorb. So imagine some gigantic Lego construction, like you've made a giant castle. Well, that might be what a complex carbohydrate or a protein or a fat would look like. But that's way too big and complicated to get absorbed into your bloodstream. So we're going to have to break apart your Lego castle into the teeny tiny single Lego block. So it's broken down chemically into these smaller blocks, and then they can make it into the vein. So these enzymes will work only on their specific substrate. And if there is no substrate, you can put all the enzyme in the world, but no reaction will happen because it's kind of like that lock and key thing. It only works on a specific substrate. Most of the names of the enzymes are going to end in ASE, and whatever precedes ASE will tell you what the enzyme will work on. For example, lipase breaks down lipids. Proteases break down proteins. Amylase in general works on carbohydrates. Now there are many very specialized proteases, lipases, and carbohydrate enzymes. We're going to just learn the basic levels of those. Enzymes work only on one substrate, but they do need to have an optimal temperature and pH. So we'll find one protease that will not work unless the pH is well below 2. So you can have a basic environment, alkaline environment, nothing's going to happen. So that we have to meet some specific conditions. Enzymes are secreted into the lumen of the alimentary canal to go find their substrates and work on them. They are not secreted into the blood, and they do not travel to other organs like a hormone. Okay, for carbohydrates, we have 3 different forms of the large polysaccharide. So the polysaccharide is the carbohydrate Lego castle, if you will. And it comes in 3 forms. 1, glycogen. This is the stored form in animals. So when we store carbohydrates in our liver or in our muscle, they are stored as glycogen. Likewise, if you eat the organ or muscle of an animal, you will get a little bit of carbohydrate. Starch is how we store carbohydrates in plants. So when you eat starchy foods, you are eating the plant carbohydrate. So that's potatoes and celery and apples and all things like that. Cellulose is the other form, again, a very large polysaccharide that is what we call fiber. And this is the cell wall of plants that we are unable to digest and get any calories from. Sometimes it's called roughage or bulk in your digestive system. So salivary amylase that we have seen is produced by the salivary glands in the mouth is going to act on some simple carbohydrates, and they will be broken down. You know this happens because if you lay a cracker or cookie, a piece of bread even, on your tongue and just let it sit there, mix with a little saliva, it's going to break down and change, right? And dissolve. So that's salivary amylase in action. There are no proteases and there are no lipases in the mouth. So you could put a piece of steak protein, right, on your tongue. You can coat it in all the saliva that you want, but nothing is happening to break it down chemically, so it's not going to change. So salivary amylase can just work on the very simple carbohydrates in your mouth. Food goes into the stomach. No carbohydrate digestion goes on in the stomach. And then once we reach the duodenum, we will have access to some pancreatic amylase. So that pancreatic juice made by the pancreas is going to have some pancreatic amylase in there to break down the disaccharides, break down into disaccharides. So break down these bigger carbohydrate molecules into disaccharides, which are two saccharide or two sugar molecules. And these disaccharides that we're going to talk about are lactose, maltose, and sucrose. Lactose you are familiar with as milk sugar. Sucrose, you may not be familiar with that term, but you know what sucrose is. Sucrose is table sugar. And maltose is found in grains. And then, your digestive juices are going to mix, they're going to break them down, and then further down your intestine, when we reach the brush border of the small intestine, those enzymes will further break the disaccharides into a monosaccharide. These are the building blocks of carbohydrates. The monosaccharides are the forms of carbohydrate that can be absorbed into the hepatic portal blood system. And these are, well, we talk a lot about glucose, but also fructose. Fructose is the same recipe as glucose, just a different shape, and galactose. This is a picture showing you polysaccharides, disaccharides, and monosaccharides. So you will need to know this. Like I said, we talked about this in chapter 3. We will also talk about this in chapter 24, which is the nutrition chapter. But when we're talking about digestion, I want you to understand what each enzyme does and why we have to have multiple enzymes to break down the carbohydrates. So again, imagine if you have your Lego castle, if you just kind of like bump it over, it's going to break into a few smaller pieces, but there's still going to be a lot of pieces that are connected. So if you bump it over, that's the first amylase right here. And then if you take your hands and kind of like pull them apart, you'll get some smaller pieces. That'll be like these breaking these other bigger saccharides into the disaccharides. And then if you sit and individually pick apart the other resistant pieces into single little Lego blocks, that will be the action of your maltase, your sucrase, and your lactase, breaking things into the monosaccharides. And we have to get to the monosaccharides because the small intestine can only absorb monosaccharides. All right, protein. So we're going to see the same kind of thing happen. We'll have the giant protein, which is your big protein castle. And then we will have the beginning of protein digestion is going to be in the stomach. And is going to be by an enzyme called pepsin. Then the larger protein fragments are going to be broken into smaller peptides by some other pancreatic proteases. So protease is going to be the big generic name for enzymes that digest proteins. There are some specific ones, trypsin and chymotrypsin, that you should know about. And then finally, in the brush border, we'll have brush border proteases that will break the short peptides into single amino acids that can be absorbed. Fats are usually found in the form of triglycerides in foods. And these are, triglycerides like to hang together and clump together in big fat blobs. So imagine that the big flat blob is the equivalent size of like a beach ball of fat blob in your digestive system. There's very little surface area and there's a lot of fat around it. And it's very hard for the lipases to act on that huge surface area. So before the enzymes can really work, we've got to break the big blob apart into smaller blobs. And we do that with bile. So bile, remember, is made by the liver, stored by the gallbladder. And when we have a meal that has a decent amount of fat in it, we're going to release some bile to help break it apart. So bile is going to break the big blob of fat, like the big beach ball size blob of fat, into smaller, like golf ball size balls of fat. So we have much more surface area for the lipase to be able to work on. Think about it also, if you've ever watched a commercial for Dawn dishwashing detergent, if you've ever washed dishes, you know that we use a soap detergent to break down, especially the fatty, greasy stuff, so it's easier to clean. So you could also say that bile works as a detergent. Fat will then be digested. So it's important to know that bile does not digest fat. What the word we use is emulsify. So it breaks it down into smaller pieces. And then once the fat has been emulsified, it can be digested by lipase. And lipase, there is some lipase in saliva, but it doesn't really get a chance to do its job until we make it all the way down to the small intestine. So fat digestion predominantly occurs in the duodenum. That's what I want you to remember. And we're going to break down triglycerides into fatty acids and glycerol molecules.